Have you ever thought about the fact that you're made of trillions of tiny factories? I mean, microscopic powerhouses, each one working 24/7 just to keep you alive. Well, today, we're going to shrink down and take a tour of these incredible little engines, the cells that build every single living thing on Earth. You know, it sounds like something straight out of a sci-fi movie, doesn't it? But this invisible universe is absolutely real. And for most of human history, it was completely hidden from us. Unlocking it didn't just change science, it completely flipped our understanding of life itself on its head. And the key to this whole hidden world? Yep, a single tiny cell. So our story starts with just a handful of brilliant pioneers. And what's so fascinating is that this wasn't some single eureka moment. No, it was a slow, really painstaking process of discovery that stretched out over nearly 200 years. But it eventually pieced together one of the most fundamental theories in all of science. All right, so let's meet the very first detective in our story, the guy who first peeked into this hidden world and ended up giving its most basic unit a name that stuck. So back in 1665, a scientist named Robert Hooke pointed his pretty basic microscope at a super thin slice of cork. And what he saw looked just like a honeycomb. These tiny empty boxes reminded him of the small rooms or cells where monks lived in a monastery. And just like that, the building block of life had its name. But here's the thing. Hooke was only seeing the empty shell of life, right? The leftover walls of dead plant cells. To see actual life, living, moving cells, science needed an unexpected hero. Not some famous professor at a university, but a Dutch cloth merchant named Antony van Leeuwenhoek, whose secret obsession was crafting the most powerful microscope lenses the world had ever seen. And get this, Leeuwenhoek's handcrafted microscopes were true technological marvels. We're talking up to 50 times more powerful than anything else available at the time. So when he peered through one at a single drop of pond water, he discovered an entire universe teeming with tiny, swimming creatures he called animalcules. For the very first time in history, a human was actually witnessing the bustling, hidden world of bacteria and protozoa. Incredible. So these first glimpses were, absolutely mind-blowing. But it took almost 200 more years for scientists to really connect all the dots. They'd seen the pieces of the puzzle, now they had to figure out the rules that governed this entire microscopic universe. And those rules became what we now call the cell theory. And let me tell you, this is the foundation of all modern biology. It's really simple, but totally revolutionary. 1. All living things are made of cells. 2. The cell is the most basic unit of life. And 3. Every single cell has to come from another cell that already exists. Of course, science is a human story, right? Which means it's full of rivalries and forgotten heroes. While a guy named Rudolf Virchow usually gets the credit for that third rule, it was actually another scientist, Robert Ramack, who first published the evidence for it. It's just a powerful reminder that history doesn't always tell the full story, and that scientific progress, well, it often comes from a mix of collaboration and even a little bit of controversy. Okay, so now that we know the basic rules of the game, it's time to step inside. We're going to tour the cell as if it's a complex, high-tech factory and check out the incredible machinery that keeps the whole operation running. First, a quick heads up. Not all cells are built the same. You've got your simple prokaryotes, like bacteria. You can think of them as a basic one-room workshop. But then you have the eukaryotes. These are the complex, compartmentalized cells that build plants, animals, and, well, you. And these are the sprawling, sophisticated factories we're about to explore. So inside each of these factories, you have specialized departments called organelles. Think of it this way. The nucleus, that's the head office, storing all the DNA blueprints. The ribosomes are the workers on the factory floor, reading those blueprints to build proteins. They do their work on the endoplasmic reticulum, which is like the main assembly line. And once a protein is built, little transport carts called vesicles carry it over to the Golgi apparatus. That's the shipping department. This whole protein production line is just a masterpiece of efficiency. A protein gets built on the assembly line, it's packaged into a transport cart, and then it's sent directly to the shipping department for final processing and shipping. It's a perfectly organized system, and it's happening trillions of times a second inside you right now. Just think about that. Now, let's zoom in on what I find to be one of the most fascinating parts of the whole factory, the Golgi apparatus. Honestly, calling it a shipping department doesn't even do it justice. It's more like a highly advanced, intelligent post office. So the Golgi looks kind of like a stack of flattened pancakes. As proteins arrive from the assembly line, the Golgi gets to work.
It modifies them, maybe adds a molecular shipping label or tweaks their shape. Then it sorts and packages them for their final destination, whether that's somewhere else inside the cell or outside the cell entirely. But this all raised a huge question for scientists. How does the cargo actually move through this post office? I mean, what's the mechanism? It's a puzzle that sparked a major scientific debate, leading to two completely different theories about how it all works. Okay, so the first theory is the mail cart model. In this one, you imagine the Golgi's pancake sacks are stationary and tiny vesicles act like little mail carts, carrying proteins from one pancake to the next. The second theory is the conveyor belt model. Here, the pancakes themselves actually move, carrying the proteins along as they mature, before the whole thing breaks apart at the very end. And this debate is super critical, because when this cellular post office makes a mistake, it can lead to devastating diseases tied to misfolded proteins like cystic fibrosis. It just goes to show you that even now, the cell still holds deep secrets we're working hard to uncover. So we've gone all the way from the discovery of the cell to these intricate debates about its internal machinery. But why does all this tiny stuff actually matter? Let's zoom back out and connect these microscopic factories to our own world. And the reason it all matters so much really comes down to one extraordinary type of cell, the stem cell. A stem cell is like the factory's master blueprint. It's a completely blank cell, a clean slate with the absolutely incredible potential to become, well, pretty much anything. And that potential is just staggering. A single stem cell can be instructed to become a beating heart cell, a firing neuron in the brain, or a filtering liver cell. Scientists are already harnessing this power to do amazing things, like regenerate damaged tissues, grow new organs for transplants, and even create meat in a lab without ever harming an animal. This isn't science fiction anymore. This is the future of medicine. So from a monk's room in a piece of cork to a factory assembly line to the future of medicine itself, the story of the cell is really the story of life. It leaves us with one final incredible question to think about. If we truly are made of trillions of these tiny, brilliant factories, what amazing things are we going to build with them next?